Ken Carter. Oh, there you are. Hi, Ken. So listen, I had a chance to talk with Ken briefly yesterday, and I was actually very, very excited in our conversation. NexImmune is a company that, I don't know that it's correct to say that you were spun out of Johns Hopkins, but there are a lot of deep roots associated with Johns Hopkins, which I would consider to be one of the finest medical research institutions in the country. But in my conversations with Ken yesterday, you hit upon one of my favorite topics, which is, what should the vector be? When we talk about CAR T therapy, if we talk about dendritic-based cell therapy, the question is, what's the right carrier? And so what got me very excited in the conversation with Ken was that NexImmune is really looking at a pharmaceutical-like solution to that, really taking almost the biology out of the carrier. And I think in that way that this is maybe some of the most cutting-edge science that we could see. So don't be fooled by the fact that it's late in the day here and that you've seen lots of other presentations, because while this is a private company, there is a very, very significant list of intellectual capital associated with this company. And I would tell you the technology itself is very, very exciting. Ken, will you introduce it to us?
Well, that's great, Jason. And thank you to Jason and the organizers for giving us the opportunity to step in here. To actually follow on to one point you made, I'm going to show just a couple of slides. And one of the things I hope people will come away with is not only is this the very best and very cutting edge science we believe in this space, it's actually the science we believe that's going to be most directly translatable to, as you say, a carrier that not only has all of the benefits of a pharmaceutical, but also, like many of the other companies here, can really take advantage of many of the cellular, the opportunities of a cellular therapy. So the next, do I switch this up here? Is that? Yep. So what I'm talking about is a technology that we spun out of Johns Hopkins, which we've labeled the AIM, or artificial immunity technology, which is based on a totally artificial antigen-presenting or dendritic cell, which you see in the upper right of this panel. And we think that this technology will provide the opportunity to really bypass or surpass many of the challenges of many of the cellular approaches. I'll talk about that on the next slide, and it has the opportunity to really take advantage of the great progress that we've seen, driven primarily by the various checkpoint inhibitors. But beyond what the checkpoint inhibitors have done, we think both as a standalone treatment and in combination with the checkpoint inhibitors, we think we'll be able to go to many, many more patients, more tumor types, and in many cases where the checkpoint inhibitors could actually be better or get to more patients, we think this is a technology that will allow them to do that.
But before you advance, Ken, I just want to make it very clear to everybody, because it took me a couple of takes to make sure I understood what that picture is in the right.
Should we go to the next slide?
Yes, please. Great.
Yep.
Thank you.
Yeah, absolutely. So just to be clear for everyone, even though we call this an artificial antigen-presenting cell, it's actually not a cell at all. It's a pharmaceutical product that's based on a nanoparticle matrix to which you have attached the classic signal 1 for a dendritic cell, which is the presentation of the antigen in the context of an HLA molecule, and classic signal 2, which is a direction to the T cell to either be up or down regulated. We're in the fortunate position that there's a very deep IP portfolio and many, many clinical, preclinical proof of concept studies that have been published. And the exciting parts of the technology is that unlike a typical cellular therapy of this type, there's absolutely no requirement for engineered cells, whether it's on the dendritic cell side or the T cell side. The T cell stimulation and expansion that you get is a highly polyclonal response. By the way, you don't have to just use one antigen. You can use a variety of antigens. You can tailor your antigens to individual tumors. You can even tailor them to individual patients. There's also a variety of types of evidence showing that the T cells you get are highly cytotoxic and highly functional, even beyond what you would get with a normal dendritic cells. And most importantly, and this is in the context both as a standalone treatment and as a complement treatment to checkpoint inhibitors, this technology really drives the formation of those key tumor-infiltrating lymphocytes that have really become kind of a holy grail of really expanding cancer immunotherapy. So rather than go into a lot of data, I mean, there's a lot of published data out there, and I hopefully will have the opportunity to talk to a number of you afterward, and I can point you to some of that. I just want to talk specifically about what our strategy is as a company. Our first product, which will be a direct injectable that's shown at the top of this slide, we expect about a year from now to file an IND. We should have dosed a number of patients by the second half of 2016. There's also the opportunity for adoptive therapies where you would take the T cells out of the patient, stimulate them, and put them back in. And one of the most exciting things is what you see both on the bottom here, which is the opportunity diagnostically to expand T cells on a patient-by-patient basis. And then, for example, in the case of AIM-101, our first treatment, you could, in a patient-by-patient way, say, does this patient have the immunocompetence to respond to this particular antigen? Do they have the T cells that can respond? You can very quickly get a read on that, and then you can have a customized, tailored treatment, whether it's a shared tumor antigen or a totally novel neoantigen that is specific just to that patient. So finally, I'll just close with just pointing out, as Jason said, we do have the benefit of, I wanted to show the slide because a lot of these names are not on our website yet, but we've benefited from really some tremendous guidance and continue to benefit from that, not only from Johns Hopkins, but others, as I'm sure many people will recognize some of the names on this slide. The great thing is these aren't just names. Everybody on this slide is pretty intimately involved in the company and helping us really think through sort of the strategic and clinical opportunity.
So Ken, why don't we go back one slide just so we can have that graphic up as we're talking. So what you're really saying here is that rather than using a dendritic-based vector, rather than using an autologous T cell or an allogeneic T cell, you're saying, hey, we can skip all of that. We don't have to deliver a plasmid and electroporate it into the blood. What we're going to do is deliver a platform, a gold nanoparticle, a microparticle. A pharmaceutical, and you're basically going to pull down from a menu any construct that you're going to load onto that particle that then is going to be taken up by direct.
Won't be taken up. So that's that's a very key point. Thanks. So I want to point out here, you are not dependent on either the healthiness or the function or the flavor of the individual of the endogenous antigen-presenting cells. So, for example, in the case of the suppressive antigen-presenting cells in the tumor, those don't concern you. You're going directly to the T cells. You're stimulating those T cells directly. The body experiences these artificial antigen-presenting cells as full-blown dendritic cells that directly stimulate the T cells.
So in other words, for the body's purposes, it's the same thing as a dendritic presenting antigen to the T cell that then elicits an in vivo T cell response. And what I heard you saying is that with diagnostic markers, you can determine exactly what the balance of the state of a person's immunity is, T regulator cells, T effector cells, so that you can know how to prime that element to elicit the T cell response that you're after.
Yeah, so it's not quite that. I mean, we will eventually get to that point in second generation. So in second generation, we'll be able to have cocktails that target different T cell subtypes. What we can do, we believe, in sort of generation 1A and probably as early as phase 2 trials for our first product, AIM-101, we'll actually be able to load the artificial cells with specific antigens, whether they're shared tumor antigens or potentially specific antigens for that patient. There's already the proof of concept on the preclinical level. By the way, loading the antigens also is just a pharmaceutical process, doesn't involve any cells, doesn't involve kind of manipulation of things. It's literally simply a chemical reaction that takes less than one afternoon. So you can have the unloaded artificial cell, you load it up after you've already tested the patient's the patient's T cell population to know exactly which ones to use.
So from the world of antisense therapy, I've learned a lot about delivery platforms and nanoparticles and technology. I've seen companies like Techmira do that. Help me understand, what was the intuitive leap that you came to in order to achieve this versus the rest of the world that's really chasing a cell-based approach?
Yeah, so I would love to take credit for that. That wasn't my intuitive leap. That was John Schneck and Matthias Oke, who are our scientific co-founders at Johns Hopkins. It was actually Matthias who came to John's lab from Germany, who had actually spent 5 years of his life in exactly that world. He had exactly been doing T-cell engineering through a variety of techniques, including RNA expression, to try and get dendritic cells to behave in a way that was amenable to treatment. He knew about some work that John had done where he'd been linking various things to nanoparticles. He came to John with the idea and said, if you could show that, if you could do the experiments to show that what I just showed on the previous 2 slides could work, and you could solve the manufacturing issues, you would completely bypass the whole world of cellular engineering in this field. And that was, a few years ago, and then we helped them spin the company out.
I don't know if you saw the CAR-T panel, but one of the things that I thought that was exciting about the CAR-T panel was the idea of engineering Boolean logic expression. And is that something that you're thinking about, where you would actually engineer a particle that would say, if this is present and this is present, but not this, then you're primed to go?
Absolutely. I didn't see the panel. I'm very familiar with the concept. This, again, on 2 levels, both the level you brought up earlier, which is which particular T-cell populations do you want to stimulate and or suppress, and on an antigen, on sort of sets of antigens, what is the profile or fingerprint of antigens that is most likely to get a response in this patient? It's absolutely amenable to that, yeah.
So tell me a little bit about what are the strategic plans of NexImmune. You've been a private company. How much money has been invested in the company today? What is the next round? Would we call that an A round? And help me understand, at some point, will this be a public company?
Yeah, that's a great question. So first of all, on a strategic level, the financing aside, the company, we're building a company. We've already got a couple of tremendous people on board, including Scott Carmer here, who is our Chief Operating Officer that joined us from a very senior position at MedImmune. We strategically will build the capability to do the things that you see on this slide. We will take the technology all the way through clinical trials, and the plan is to actually market the products that we develop. Right now, we're very focused on what you see on the top half of this slide, which is developing the first injectable product. In terms of financing, we're in the fortunate position that we raised a few $1 million in sort of get started capital from NEA, Pfizer, and Amgen last summer, and we're in the process of raising a Series A. We're an early stage company. In most markets, I wouldn't say we have our sights on an IPO, but I would say in this market we aren't paying attention.
Well, you have to pay attention to some of the valuations you're seeing in the CAR-T space, right? And if I'm sure it's not this simple, but if it's this simple and we could engineer biology out of the initial construct, that's got to be very attractive to the world of big pharma. So I would think that you probably have a lot of active BD type discussions.
Yeah, and I think not by accident that 2 of our first 3 investors are top 10 pharmaceutical companies. So
okay, terrific. Thanks. Very exciting stuff, and it's going to be worth watching.
Great.
Thank you, Ken. Thanks for joining us
Jason.
today.
Yeah, thanks a lot. Yeah.